Currently, the immunohistochemistry is used with increasing frequency to identify the presence of specific molecular markers and their changes in case of pathologies, giving also the possibility to perform quantitative analysis. The immunohistochemistry, mainly the immunofluorescence technique, is used in the larval and adult zebrafish animal model, but little is known about a good standard immunohistochemistry protocol for zebrafish. Here we describe and illustrate a protocol that can be used to study the evolutionary conservation of important proteins in adult zebrafish. A good protocol for immunohistochemistry in adult zebrafish can help to underline the usefulness of this animal model to study the morphological expression and distribution of highly conserved biomolecules. This method can also help to analyze possible alterations of these biomolecules due to pathological conditions correlated to human physiopathology. Demonstrating the procedure will be Dr. Lea Tunisi, a PhD student from my laboratory.
To begin this procedure, transfer the prepared frozen tissue blocks to a cryostat at -20 degrees Celsius. Cut the block in coronal or sagittal sections of 10 micrometers. Then, collect the tissues in alternate serial sections onto adhesive glass slides suitable for immunohistochemistry and store them at -20 degrees Celsius until ready to proceed. First, use a solvent-resistant pen to demarcate the tissue area on the slide. Rinse the sections 3 times for 5 minutes each